So we get to start now with my favorite subject, genetics. Here is a preparatory activity with a few questions. You might pause this and look at these questions and see if you already know the answers for them. Who is Gregor Mendel? What did he do? How does a dominant gene differ from a recessive gene? And then this third one's not a question, it's more of a can you do this? Draw a Punnett square to show the probability that two heterozygous individuals might have a redheaded child. And for this, we'd call red recessive. We'll talk later about how that's not exactly, but a lot of examples use that. So genetics is a branch of biology concerned with heredity. The units of heredity are called genes. Genes reside in the DNA, and the DNA is organized into chromosomes in the nucleus of the cell. These things we have already talked about as part of our chemistry chapter, chapter 2 back at the very beginning, but recalling these definitions and terms to you. Some new terms, dominant and recessive. A dominant gene will mask a recessive gene. Okay, that's just if there are two traits and both are inherited in an individual and one is shown and the other one is not, then that's referred to as masking. The one that's being shown, the one that you can observe, is masking the other one, and the one that is doing the masking is referred to as dominant, and the other one is referred to as recessive. When we talk about these in genetics problems, and I'm sure you've seen this in the lab already, in the Punnett squares, we use capital letters to show dominance and small letters to show recessiveness. So we have some terminologies there as well with homozygous. Homo means same, and zygous has to do with the zygote. What does the zygote contain? The zygote, of course, is when an egg and a sperm unite, it forms a single cell called a zygote. And in us, I know I've mentioned this before, us, plants, animals, anything that's multicellular, that single cell then begins to divide to make multiple cells, which eventually will differentiate into different tissues and organs. But to start with, you have that one cell and it has the entire blueprint of that organism. If it contains two of the same type of alleles, then that would be homozygous. Okay, so for example, here we have the two capital B's, BB. Okay, capital B, capital B. That's homozygous for the dominant trait. And we have two lowercase b's here, homozygous for the recessive trait. Okay. So when you say something is homozygous, you must be clear about whether that is a homozygous dominant or homozygous recessive you're talking about. You don't just say homozygous. However, hetero means different, so this means the zygote has different alleles, okay, one of each type, and that would be shown here, capital B, little b. So heterozygous doesn't take any other word descriptor with it. You don't ever say something is heterozygous dominant or heterozygous recessive. If you do that on the test, it will be wrong, okay, because heterozygous is heterozygous. Okay, only the homozygous has to be determined as to which form you're talking about. So Gregor Mendel, to answer one of those first questions, is referred to as the father of genetics. He was well, usually referred to as an Austrian monk. He actually grew up in was modern day Czechoslovakia. And he was a son of an apple farmer. He was able to go to school and eventually wound up going to university. He chose to go into the monastery and um, became a teacher at a kind of a high school type school, what we'd refer to as a high school anyway. Uh, and he taught mathematics and Greek, believe it or not. Of course, I've pointed out there's a lot of Greek in science and even more so in math. But that was what he taught. He never taught biology. He wanted to teach biology. He was never able to pass the state tests for teaching biology. Interesting thing though, I mean, he was a mathematician, and what he did is he took probability and lots and lots and lots of data and determined what was going on inside the nucleus of P cells, okay, before we had a microscope that was strong enough to see the stained chromosomes actually doing that. Okay. And it's pretty amazing, really, what he was able to do. But the estimates are he probably grew tens of thousands of pea plants over the course of maybe 8 to 10 years before he wrote his two papers. His first paper, he presented what was called the principle of segregation, became known as Mendel's first law. And what this paper says, what this principle of segregation says, is that two factors of a characteristic segregate or separate during the formation of eggs and sperm. Okay. That paper was published in February of 1865, and the next month, in March of 1865, he published the second paper, The Law of Independent Assortment. And that says that factors for different characteristics are distributed to reproductive cells independently. It became known as Mendel's second law. And you may be looking at those going, I don't see how that is saying anything different. Okay. So let's look at that closely. What he did was he belonged to a uh, organization that was a academy of science. There are academies of science all over the world. There are regional academies of science. There are national academies of sciences. We have a National Academy of Science here in the United States. We also have an Academy of Science here in Oklahoma, the Oklahoma Academy of Science, of which I have been a member, and I have actually had two abstracts in my life published in their proceedings. So if you present a paper to the Academy of Science, the paper or the abstract, depending on how much room they've got in their publication and how much space they want to give your information, gets published in those proceedings. So that's what he did. He presented these in two different monthly meetings. They used to meet monthly back then. Some still do. The Oklahoma Academy of Science meets once a year. Uh, so uh, he presented to those meetings, and they put his papers in their publications. Those publications are then shared with other academies of science around the world. The only reason we know that Mendel did this work is because of that, the publication in those academy proceedings. Later in 1910, somewhere between 1910 and 1920, in the teens there, his work was found by two other scientists who you have probably studied about in one of the labs already. And that was where his work was rediscovered, because it was totally forgotten. Nobody at the time really cared, okay, about inheritance and pea seeds. All right, but what this first law says, two factors of a characteristic. So he looked at 7 different characters in pea plants. He looked at seed shape, seed color, flower color. That's 3 of them. He looked at whether the flowers were on the end of the stem or off to the side of the stem. Looked at like plant height, and there's things I'm missing, I'm sure. Anyway, uh, so let's take flower color, for example, here. He's saying that if the flower is, if you've got two factors, you can have purple flowers, you can have white flowers, then those two alleles that code for that will be separated, okay, during the formation of eggs or during the formation of sperm. You've done the mitosis meiosis lab, so you know how meiosis works, and this is meiosis. So you see how the two copies of the chromosomes separate, duplicate, separate, duplicate, and we wind up with 8 in the end, okay? Uh, four, actually four nuclei. So that's where you are looking at here. Those things separate, okay? This is where we're looking at, okay, well, if a plant has purple flowers and yellow seeds, but it also carries the characteristic for, let's see, it's yellow dominant. I may have got that backwards anyway. White flowers and green seeds, then if it inherits, if it passes purple color onto one offspring, does that offspring also have to have the green seed, or can it have the yellow seed? And the deal is it could get either. Okay, it's a 50-50 coin toss on whether it's going to get purple or white if it has both alleles, if it's heterozygous. And it's a 50-50 coin toss on whether it's going to have green seed or yellow seed. if it's got both alleles, if it's heterozygous. Okay, obviously, we'll get into Punnett squares on this. If it's homozygous, then it's going to pass that one allele type onto all of its offspring. But looking here at heterozygous, and he determined all that mathematically. So here is an example of seed shape, smooth versus wrinkled. Smooth is dominant, wrinkled is recessive, designated by our capital letter versus lowercase. If we take a parent plant, that's what the P stands for, and we cross a homozygous dominant with a homozygous recessive parent. So we've got two parents here, homozygous dominant, homozygous recessive. This parent can only pass on the dominant allele to its offspring. F1, F stands for filial, means child in Latin. Okay, so this is the first filial generation. This can only pass on the dominant trait. This parent, the homozygous recessive, can only pass on the recessive trait. So all offspring of this mating are going to be heterozygous, 100% heterozygous. Okay, no other possibilities. Now, do I go on with that? I think usually I go to the board at this point. Okay, I draw out a Punnett square saying, well, now what if we took that first filial, and since we're dealing with plants, we can cross siblings with each other. We took two P's from that first parent, first filial crossing, and cross them, big W, little w, big W, little w. Okay, let me see if I can do this without causing things to. Draw my Punnett square right here. It's not going to be pretty because I'm not used to drawing with this. Okay, so here is my capital W, here's my little w. Because these two letters look very similar when you handwrite them, I try to point my capital letters and round my smaller letters on the bottom. And every time you do that with letters that look the same, capital and small, you'll need to do some type of distinction, like S's or one that's kind of a bad one. So for that, I'll make the big one a dollar sign and then the little one not. Because when you get in a hurry doing these Punnett squares, it's easy to make a capital smaller than you think and a small to bigger than you think and get in a hurry. So this is one parent, one allele here, one allele there. Now the other parent, so let's see. It's gonna be kind of difficult with the draw tool. There we go. That's what we're crossing. So I've taken this parent, put it up here. I'm bringing this parent, put it here. That's one allele. Here's the other allele. And then this is like a multiplication table. We're going to put this allele in both of these squares in this row and this allele in both of these squares in this column. So doesn't really matter what you start with. This one will come down here and this one will come across. Okay, this one will come down here. We always put the capital letter of the pair first. Okay. All right. Okay. All right. And then here, this will come over here and this will come down here. All right. So now we no longer have 100% heterozygous. Instead, we have a ratio here of one of these. Okay. And we have two of these. And one of these. Okay. So this is referred to as a 1-to-1 ratio, though if you read this correctly as a ratio, it's a 1-to-2-to-1 ratio. But we kind of shorten that as 1-to-1 ratio. You're going to always see this every time you cross two heterozygous parents. Okay, you're always going to get one of the homozygous dominant, two of the heterozygous, and one of the homozygous recessive. This can also be written as 25%, Yep. Come on, let go. It's pin's dragging. Okay. 50% and 25% of the, getting really small there, homozygous recessive. Okay. So 100% adds up to 100%. The whole is equals 1. If you've done your Hardy-Weinberg equilibrium equation, you kind of know that. Okay, that was one of the labs. Population genetics, the human genetics, I believe, addressed the Hardy-Weinberg equilibrium equation. And all of that equals the whole, okay, the 1. So, all right. Um, I think that's all I usually talk about in relation to this slide. So let me, I'll go into the next slide, but I'm thinking I have to erase this thing first. Erase all this. And I've had trouble in the past going on from getting my annotator back and getting my slide presentation back. So let me see if I can undo that and move forward with my slide presentation. No, doesn't want me to. Let me see if this works. I'll just go out and come back in. Yay, okay. 2 more terms. Phenotype. This is a description of what the trait looks like. Was that seed smooth? Was it wrinkled? Is it green? Is it yellow? Is that flower purple? Is it white? Those words are our phenotype. That's the description. The genotype is the term we use when we're talking about the letter designations or whether we're looking at dominant recessive, the homozygous dominant, the heterozygous, homozygous recessive, whatever we're talking about on that. The genotype, that's the genotype. Okay. All right. So know those 2 terms. Traits, characteristics are inherited. The gametes are the sex cells, that's your egg and your sperm. Now, flowers. Okay. Pea flowers that Mendel worked with and most other flowers, though not all, have both male and female gametes. They produce both. And I believe I've given you a really brief rundown on the flower model in the classroom, where we have the uh, stamen and the pistil. Okay, the pistil is the female part, produces the egg, the gametes, and then the stamen um, have the anthers, the top, the pollen grain, and the sperm is inside that. Okay, so that was a complete flower, had both parts, and pea flower looks like that to some extent. Your some of your like fruit trees and things are incomplete. They have what's called incomplete flowers. They either have the male parts or the female parts. They don't have both. But pea flowers do. Some more genetics terms here. We have hybrid, and this is the offspring of parents that have different forms of a trait. So back up here to this slide, this F1 generation is a hybrid for seed shape. Okay, it has one allele for smooth and one allele for wrinkled. So that makes it a hybrid for that trait. Tall versus short's another one given here. Monohybrid, this is where the 2 parents differ in a single trait. Typically, they may differ in more, but we're only following one trait. We're only interested in our breeding program in one trait. We also have other terms that we'll get to later of dihybrid and trihybrid and things like that. I probably won't talk about trihybrid. And of course, the Punnett square is the diagram used to determine the probability of the offspring types, which I just did a Punnett square for you. I believe you've been exposed to that in the labs a bit, so I don't know that I need to go over a lot of that. I will direct you to a genetics worksheet that I have for you to practice with. The answers are on that sheet for you. The first part gives you alleles of a parent and asks you to list out what alleles they could pass on. And those are the bold face letters off to the right of each of those. Those are the traits. Those are the specific alleles that those parents could pass on. Then after that, you're given some genetics problems, and what you see is the handwritten Punnett squares. And there's a little shorthand version of the Punnett square. Let me see if I can do that for you. because that can be a little confusing to you. So if I'm going to cross, um, if I'm going to cross a parent that is, to stick with what we were doing earlier, let me draw my Punnett square out here. Kind of draw it. I'm going to run into my words on this one, but okay. So if I'm crossing two heterozygous parents, I mean, oh, sorry, two homozygous recessive parents, okay, every single square here is going to be this, right? So do I even need to fill these out? They're all going to be the same. So that's what, oh, see, it takes it away in the way okay, so that's what the little shorthand squares are trying to show you there on the uh, homework sheets. Is that if both parents have this allele and uh, if one parent's homozygous for that, then you only need to draw one. If parents are homozygous for that, you only need to draw one. Generally speaking, it's something more like this being shown, okay, where you got a parent that has heterozygous being mated with a homozygous recessive. Um, this square down here, if it were here, would be identical to that. And the square down here would also be identical to that. So why mess with duplicating the second row? What I recommend to you is draw out the full Punnett square. Okay, I did not make up that worksheet. I inherited that worksheet from the instructor previous to me. I really like it, so I didn't want to redo it. I just took it and have been using it for years. Um, but for me, I would recommend to you draw the full Punnett square every single time. That way you don't make mistakes, don't take shortcuts. But why I left it on those sheets and didn't redo that is that if you see the one square, you know all four squares are the same. If you see two squares, you know that those two squares are repeated. Either the next column over or the next row down is going to look exactly like that. Okay, however it's laid out. All right, so you can check yourself with that. And then the actual answers are in boldface, typed boldface underneath the handwritten Punnett squares. So the Punnett squares are just used to find the answer, to find the ratio, like I was showing you previously, the 1:2:1 ratio, or whatever ratio you want it being, because depending on the cross, it may be a 3:1 ratio. It may not be the uh, the 1:1 ratio, or 1:1. So the Punnett square is just a tool to find the answer. It is not the answer. And that is something you need to remember on the big quiz and on the test, the final at the end, is that the Punnett square is hardly ever the answer. Okay, the Punnett square is the tool you use to get to the answer. So the answers are actually typed out in boldface underneath. So cover up your answers, work them on your own, and then check yourself. And there's a lot of practice on there. There are also dihybrid cross questions, which you won't be able to answer until I talk about that. Right now, we're just doing, this is just a monohybrid, and that's usually, I think, the front and back the first page, maybe part of the second page, can't remember exactly. But I'm going to stop this presentation right here, right now, and you can go work some monohybrid Punnett square problems. Get comfortable with those. Again, I think the lab is taking you through some of that, but there's some more practice for you. And when you feel really comfortable, then we can move on. You can come back to the next presentation.